Good afternoon. It's my pleasure to be here today and discuss a little bit of what we're doing at the American Society of Clinical Oncology. This is the Society for Cancer Doctors, think of us that way. Some 35,000 doctors from over 100 countries around the world. And lately we've been thinking about what are the big needs for the cancer field. And so in 10 minutes, what can I possibly say about that? Well, one of the big issues we have is how slow we are learning. Many people have criticized the investment in cancer research as being unproductive. Why haven't we cured cancer yet? We put a man on the moon. So why haven't we fixed this problem? And I think most of you realize the problem is that we don't know the underlying scientific reason for cancer. We don't understand it. And if we don't understand it, how can we possibly do anything better than throwing rocks at the problem? So we've had this slow problem where we really didn't understand the causes of cancer. And the causes of cancer were understood from tobacco and things like that, but what actually were the scientific basis for cancer? And so we've begun to peel that back. As you know, we call it precision medicine. That's the understanding of the molecules that are going wrong in a cancer cell, the molecules being the DNA, the RNA transcripts, the proteins, the lipids, the carbohydrates, the metabolites, the immune regulation. And in the last 10 years, there's been a plethora of information coming out of labs, like many here at Stanford and at other places around the country and the world. And so we are now starting to get production through the system. The problem is that our system for taking that knowledge and developing into learning is still very 19th or maybe at best 20th century. So it's very, very slow. So the pipeline is slow, the throughput is very slow, and we're in danger of having a traffic jam, a bottleneck in trying to get that information from the laboratory to the bedside. And that's why our learning is slow, and that's what we need to fix. So how does it normally work? Well, normally a discovery is made in the laboratory. We call it translational medicine. With that insight, a drug or molecule can be developed. Then that goes into clinical trials, and that's the second part of learning. Clinical trials are done. If they're successful, the FDA approves a drug. And then, ideally, we would then optimize that drug. We would figure out how to really use it in the real world, and very often we don't do that, but that's one of the problems. So right now, there are about 1.7 million Americans who will be diagnosed with cancer this year. That should be a lot of information if we were using it properly, which we're not. Because until 5 or so years ago, all this was on paper medical charts. So they were not collected in any way that was reusable. We did have a process called clinical trials, where we were very careful to do paper documentation of records, but at least we had it written down somewhere. And that was necessary to submit a drug to the FDA. But only 3% of Americans with cancer go on clinical trials. This has been a problem we've had for many, many years. And so we have very little information, and it's not really representative of the 97% of the population who is getting cancer. So why is that? Well, again, if you don't really understand the problem, and you don't really know what your therapy is doing even, your chances of finding a successful treatment is pretty small. And one way to juice that a bit, if you will, is to be very selective about your patients. So you pick patients who are healthy, except for the fact they happen to have cancer. They have no kidney disease, they haven't had a heart attack, they haven't had a stroke, they're not on other medications, and they're more likely to be able to tolerate all the side effects. So if you look at the typical patient who's on a clinical trial, that 3%, only 25% of them are over 65, whereas we know almost 2/3 of cancer patients are over 65. They tend to be healthier than the average patient. In this study, they went back to see in the real world, we call it people who are getting the medication for kidney cancer, only 40% of them would have qualified for the clinical trial because of other health problems. And they tend to be not very diverse. They tend to be mostly Caucasians. So we really can't extrapolate our learning very well. Another problem that has developed, it's a good problem, is we realize that cancer is not a monolithic disease. So we used to think that, for example, lung cancer was lung cancer and everybody was treated with the same medicine, and response rates of 20% were considered successful. We now realize that there are molecular aberrations. We call them molecular drivers. These are, in fact, true drivers of cancer. They're not bystander-type mutations of an unstable genome. They actually are the causative agents for cancer, and treatments targeted against these molecules against these aberrations will actually make patients better. So we've begun to subdivide cancers into classifications within that anatomic diagnosis. And you can see several of the genes that are listed here that are known to be mutated. And over half of lung cancer now is associated with a particular driver mutation. However, that means that our problem has gotten more complex. Instead of treating one disease, we're treating maybe 20. And every patient is different, and we have to figure that out. Another problem is once we've got the drug, and say we've actually figured out the biomarker associated with that drug so we can associate which patients should get that drug, we still need a doctor, a nurse, or someone to give that medication. And this poor soul lying here is representative of a doctor. This was a cartoon made by a friend of mine named Dan Macy's. And he just sat down and looked at, in 2013, last year, how many citations there were in Medline. And he said, well, let's just pretend that only 1% of that was actually stuff you really need to know as a cancer doctor. And then he said, let's assume you read 2 articles a night, which I can assure you very few of us do, and 365 days in a year, so you'll read some 700-odd papers, and you would be, that's only 10% of what you should be reading. So at the end of the year, now you're 10 years behind, and next year you'll be another 10 years behind. So what do you do with this information overload? How can we speed this up? So this is where we think big data can help us, because big data deals with these problems. It deals with large volumes of data. It deals with a lot of variety in that data. That's often unstructured, and it deals with data that's coming very fast, with high velocity. And these are the characteristics of cancer knowledge at this point. So if we had big data, we could use that 97% of patients with cancer who are being treated, but right now that data is just lost to us. And the reason why we can do that is over the last 5 years, the vast majority of cancer doctors are now using electronic health records, either in the hospital or in their office. We estimate at least 70% are now using that. So if there was a way to tie all those electronic health records together into a trusted source and aggregate that data, we could begin to mine that data. So, for example, another way of looking at it, if there was one patient, we call these outlier responses. So one patient has a phenomenal response, lives 5, 10 years. Everyone else, unfortunately, dies. In the past, that drug would be thrown away. The company would say, we'll never make money on this. It's a loser. Forget that one patient. It's gone. But in fact, if you could actually aggregate a lot of patients, many of them off trial, you might then begin to find, there actually is a subpopulation out there. Maybe it's one of these driving mutations. Maybe we should study this patient, find out what's going on here. Why did they respond, and why did patients like that one respond? So this can become a powerful tool for us. Another way of looking at this is the paradigm shift in our thinking. In the way we used to think, and we still think, and it's necessary to do this, we did clinical trials. That means that we had a hypothesis. We tried to replicate a scientific experiment. So you posited in a question, you asked an answer, you designed a study, you collected patients and data, and you analyzed it, and that was a clinical trial. A lot of trial and error, and you just go back and repeat. With big data, we can turn that around a bit. We can collect the data first, that 97% of data that isn't collected in a clinical trial, and we can begin to analyze it using artificial intelligence and analytics and begin to derive insights that give us more viable hypotheses. We can then plug that into what we call adaptive clinical trial design, which is better suited to this big data, and derive our insights and compress the time for drug development. So instead of getting 3% of the data in a limited snapshot of time, we more rapidly collect a large volume of data, retrospective and forward-looking, with all the variety and all the variation that occurs in the real world. So that gets us to the point of taking an idea from the laboratory, from the bench, and bringing it into drug development. The drug is approved, but how do we help that poor doctor in there who's suffering, trying to figure out how do I keep up? So we begin to look at the big data of real-world data patients. So we take all the patients that we have in the medical records, we aggregate them together, and we begin to do what we call population outcomes. So we say, in this population of lung cancers, let's look at all the EGFR mutated patients, let's see how they did, how long there was a response, what side effects. We can begin to understand that. We can also begin to look at healthcare operations. As we are pressed to have more affordable, sustainable healthcare, how can we deliver healthcare more efficiently and affordably? And by looking at these population studies, we can do that. And then we can provide clinical decision support. which are apps, if you will, that help the provider understand what is the information they need to look at, what is the information they need to focus on, and what are the treatments that would work best. And so we begin to get a process where we are providing doctors with real-time feedback and information and giving them guidance and best practices. So we are creating a system called CancerLinq. That's what the American Society of Clinical Oncology is doing. And we have prototyped this. It was a successful prototype. I won't go into that because I'm running out of time. But we are now building a production model for this. We have lined up 15 practices across the United States, large private practices as well as 2 academic centers who have agreed to donate their data as soon as we have the platform built. We've been negotiating with various vendors after we received an RFP. We are down to 3 final vendors. We think in the next 1 or 2 months, we'll have our final selection of the vendor, begin to build the platform this year, and then populate the information, the real-world information from these 15 large institutions into the system. That will flow into CancerLinq, which is our rapid learning health system. This is a data repository. It is not a data registry. We're collecting all the data. And begin to look for the insights that we can derive from that. So I'm out of time, and I don't think I have time for questions. I'd be happy to entertain them, but I think we have another speaker that I'm anxious to hear coming right after me. I'll circulate around if there are questions later on. Thank you for your attention.